Hi everyone, my name is Ehsaneddin Asgari. I'm a postdoc at Helmholtz Center for Infection Research. Today I'm going to present the joint work of Mufrad Lab at UC Berkeley and McCarty's lab at the Helmholtz Center for COP4, where we worked on fine-tuning of language model-based representation for protein functional annotation. And this is a joint work with Andrew Dixon, Maysam Ahmadi, Mohammad Khodabakhsh, Alice McCarty, and Mohammad Mufrad.
This project is in the framework of live language processing. And this presentation is the compressed version of our longer talk at COSI function. So for more details, please refer to this longer talk. In COFA4, we were given 3 subtasks. The first task was prediction of gene ontology terms. For the training of the network, we used around 500,000 training sequences from SwissProt that they were annotated for 28K distinct categories, structured in a hierarchical manner. And of course, it's like a multi-label classification task. And the second task was prediction of human phenotype ontology terms that we used around 4,000 training instances that they were annotated for almost 9,000 distinct categories. And again, it was also like a multi-label classification task. And the third task, in which we had lower number of training instances, was prediction of disordered ontology. And we had 1,713 training instances with 45 categories, and it was mainly like a multi-class classification problem setting. As we can see, the second and the third scenarios are low resource setting that we have only few thousand training instances. So this motivates use of transfer learning from a high resource setting like gene ontology prediction or language modeling for these cases. The solution that we suggest is as follows. So we start from the self-supervised or unsupervised training of the language model using a skipgram network that we train over large set of sequences, for instance, 100 million sequences in UniProt. On the next, we use the pre-trained embedding in the skipgram network in a classification network for prediction of gene ontology. So we use the averaged embedding and we fine-tune this embedding during this training, and by getting the weighted average, we apply a sigmoid layer to predict the gene ontology classes. On the next, when we have like a low resource setting like this disordered ontology that we have only 1,000 training instances, we fine-tune the representation that is already fine-tuned for the task of gene ontology. So this way we cascade the use of pre-trained embedding and fine-tune them in different levels to achieve higher performance. Brief report on the results. For gene ontology and human phenotype prediction for the purpose of evaluation, we use multi-label accuracy metric that is defined as average of the intersection between the predicted labels and the expected labels over the union. For the gene ontology prediction, we use 382K instances for the training and 154K for the test. And as discussed, we fine-tune the pre-trained skipgram embeddings for this supervised task of gene ontology prediction. And we achieve the multi-label accuracy of 0.3 for 28K labels. For the task of human phenotype prediction, we use 3,000 training instances and 1,200 cases for the test. First, we fine-tune the skipgram embeddings for this supervised scenario, and we achieve the accuracy of 0.21 for these 45 labels. The second, we fine-tuned the network that is trained for gene ontology prediction in the previous step, and now we fine-tune this already fine-tuned network for gene ontology and skipgram, this time for the human phenotype prediction. And it increased the accuracy from 0.21 to 0.23. Another approach that we included in our submission was prediction of gene ontology terms, this time only for top 100 frequent Go terms, in each category of biological processes, cellular component, and molecular function. And we use just multi-layer perceptron as the model, and we test the performance of protein vectors versus concatenation of k-mers with the ProtVec, and we showed that we can achieve high macroresponse when we use the concatenation. And finally, for the evaluation of disorder ontology prediction, which is a multi-class classification task, we use 1,500 training instances and 200 test cases. We report both macro F1 and micro F1. The macro F1 would be more challenging as we have 45 classes and the random chance would be around 0.02. We observed that when we use the fine-tuning of the trained representation, we achieve like a boost in the performance, especially when we have like the cascaded fine-tuning of the representation. So firstly using the self-supervised skipgram and then fine-tune it for the gene ontology prediction. Using this approach, we achieve the micro F1 of 0.86 for top 5 based on the top 5 result, and 0.17 for macro F1 for all classes. And this is also higher, at least in the micro F1, when it is compared with use of ProtVec or concatenation of ProtVec and k-mers using SVM model. Conclusions. Biological sequences can be seen as sentences of the language of life. Language modeling is a general-purpose self-supervised task that can help the downstream protein informatics tasks. Self-supervised and supervised trainings can be cascaded to help a loaded resource scenario of classification. Combination of ProtVec and the k-mer representations can improve the performance. Currently, we are working on different language modeling networks that can be explored for functional annotation. Links to the publications and the code bases for Life Language Processing project can be found on the LLP website, llp.berkeley.edu. Thank you for your attention, and I also would like to thank the organizers for their great effort on making the Kappa Challenge and serving the community with this great shared task. Thank you.